We're preparing the streaming now. It should be done in a moment. We are live. Thank you. You may start at any moment.
Okay, great. Hi, everyone, and good afternoon or evening or morning or wherever you're listening in from. My name is Colby Ford, and I am a researcher at UNC Charlotte. And today I'll be talking about visualizing transmission networks using a tool that I and some other researchers created that's called StrainHub. Just a little 2-second spiel about me. Again, my name is Colby Ford. I'm a bioinformatics researcher at the Department of Bioinformatics and Genomics at UNC Charlotte. I work in the JANUS lab, and then I'm also associate faculty in the School of Data Science. I teach cloud computing and work in the data science master's program. Outside of the university, I am an AI architect for Blue Granite, which is a Microsoft partner consulting firm. So I work in the realms of cloud computing, parallel computing, and machine learning. And then inside the university, I work and apply those concepts in the realms of infectious diseases and human genomics. Today, I really want to talk about how do we start to digest information when phylogenetics gets too complicated. So since the beginning of the first case of SARS-CoV-2, I just looked at this up the other day, so there may be even more cases now, but this is a tree that you can get from NCBI of all the SARS-CoV-2 sequences. There's over 8,000 at this point. And when you have that many sequences and you put them through your lovely software and you want to make a phylogenetic tree out of that, out of those sequences, maybe that's great and maybe that's easy and digestible, but then to me, 8,000 sequences turns into this, cool looking tree, but it's not necessarily digestible as far as the other information that goes along with it. So traditionally, one of the things that I see people doing is they'll take a data set and then they'll do some stuff with the tree to help kind of gain some other information about it. So today I want to show just some examples of a smaller data set of SARS-CoV-2 sequences and SARS-like viruses. So the tree that I'll show today is 24 genomic sequences of SARS-like viruses, temporal and geographic metadata. So these sequences are bat-hosted, pangolin-hosted, and human-hosted sequences, most of which are bat and pangolin hosted that share recent ancestry from the human hosted SARS-CoV-2 sequence that we saw in Wuhan, China. One of the things that a lot of people do when they create a phylogenetic tree is perhaps you might use a piece of software like Mesquite. I don't know if anyone's ever used this lovely piece of software that I have pulled up here. But perhaps you might color the tree by a particular state in some metadata, what we call a character in the data of the tree. So here, what I'm showing on the screen is that same tree that I had before of 24 SARS-like viruses. But then I've colored or overlaid a color for the locations on a map. And maybe I want to know how this virus moves about. But maybe this isn't it. This isn't all that I want to look at. One thing that I want everyone to keep an eye on is this NC taxa here is the, or taxon here, excuse me, is the Wuhan, kind of one of the first isolates of SARS-CoV-2. All of the others are pangolin and bat hosted. And so let's say I want to understand how this virus is moving, or these types of viruses are moving throughout an area geographically. Maybe I can take a tree like this and color it. And maybe I can get some information out of this, but maybe it's not exactly clear. And then the other is maybe I want to colorize by host. So I want to see how there's host to host changes across this lineage. And so this might be a little bit easier to understand, where we've got bats and we've got bat-to-bat transmissions, no problem. We've got some bat to pangolin that we see, and then we also have bat to human here in this NC isolate here, which is the human, one of the first SARS-CoV-2 isolates. But one of the questions with this is that, how do we actually start to share all of this information kind of at once? In the Janus lab at UNC Charlotte, we've developed a piece of software called Supermap. This is an older piece of software that allows kind of a Google Map-like experience, but allows you to overlay a phylogenetic tree on top of the map. This piece of software is really nice because it allows you to kind of see the transmissions using the phylogenetic tree. But sometimes when there is a relationship that's kind of a back and forth geographically, perhaps the backbone of the phylogenetic tree doesn't exactly show you what you want to see. And so the same data I'm showing here on this right map, I have those same 24 taxa in the data set, but it's not exactly clear how all this is moving in this exact this exact visual. So most recently, we created a piece of software called StrainHub. Some of you may have heard of it. StrainHub allows for you to kind of bring your own data, BYOD, and it allows for you to bring a phylogenetic tree, some accompanying metadata, and any geodata that you want to visualize. And I'll demo this in just a second. So I'm showing the same exact data here as well. So this is an online web app that you can access at strainhub.io. And like I said, I'll demo this in just a second. But this allows for you to summarize a phylogenetic tree by whatever type of metadata that you want to upload to accompany it. So instead of looking at a phylogenetic tree where it's not exactly clear where this particular virus is moving in this area of China, or maybe it's not exactly clear how this is moving from host to host, now I can represent this same phylogenetic tree as a network based off of changes between different states. And a state can be whatever you define. In this case, a state could be a particular location, as you see here in the network to the left, or it could be a host. I do a lot of work in malaria, and we use host, we use location, we use species sometimes. There's all sorts of things that we can use there. And using StrainHub, this makes it pretty easy to visualize this information. And in addition to just visual networks, I can also overlay this network on a map, as you see here on the right. So now you can kind of see what the progression or transmission of the virus SARS-CoV-2 is in this particular area. So let me move out of this for just a second and show you StrainHub. So StrainHub has been around as a beta tool since probably end of last year, but earlier this year was published in the Journal of Bioinformatics. So if you navigate to strainhub.io, you can see this site for yourself and use it. Once you get started, you have the ability to pull in various different types of phylogenetic trees. You can use parsimony trees, beast phylogeography files, or you can bring in a aligned FASTA file, and we'll create a neighbor joining tree for you. If you bring a parsimony file, all you have to do is pull in that information. Let me get my information here. All right. I can pull in my tree file. I can pull in my accompanying metadata information. I can pull in my accompanying geodata information. The geodata is optional. You don't always have to bring that in if you don't want to visualize things on a map. If you just want the network, you can leave that out. Once I have this information, I can click the List States button, and then from the bits of information that are in the metadata, I can pick a particular state that I want to map out as the network. And then from that, I can also choose a particular centrality metric. In the paper, you can read a bit more about these. I won't waste time here with that. I'll choose source-up ratio for now, and I can generate a particular network. So instead of showing the tree as this kind of complex structure, I can kind of summarize that tree based off of the metadata that we have here. So in this scenario, you can see that we're looking at bat to pangolin transmission and bat to human, but no pangolin to human, at least in this particular data set. I can also regenerate this this network as using the state. So this is a state, particular state location in China, and you can see that Hubei seems to be a bit of the hub here, and then you can see how the virus is moving, or these SARS-like viruses are moving throughout the region. And like I said, in your data set, if you have particular other attributes in your metadata, you can add that here. You could add a component with time or whatever you'd like. It's actually quite flexible. And then from this, the software also allows you to see a tree visualization. and then a map. So you can see the kind of exact same information as the network, but now overlaid in over a leaflet map. So this is a tool that requires no coding. As long as you bring your own data, bring your own metadata files and your tree files, you can generate these for yourself. But one of the questions that I had before, and that we've had some other students that start using this, is let's say I want to look at things by location and by host at the same time. The web application is quite flexible, but it doesn't allow you to do every possible combination of everything that you have. And so the question I had before I was asked to give this talk is, why not both? ¿Por qué no los dos, right? Why can't I do multiple things at once? And while we are going to be adding more additional things into the user interface, one of the things that's kind of challenging with this is that there are so many things that can be done with these files, so many customizations, that what we decided to do is, drum roll please, as of today, we're now releasing StrainHub as an R package. So this is the beta version of StrainHub as an R interface. So now you can go to strainhub.io, you can bring your files there, you can create your maps, your visualizations, and then if there are further customizations that you want to do above and beyond what the web app allows you to do, now you can do them locally. So StrainHub uses other R packages such as VizNetwork and Leaflet to create these visuals. And now using the R package kind of functionality, you can customize to your heart's desire. So for example, also one comment here is this is under construction. This is something that we're continuing to add functionality to, both to the website itself, the web app itself, and the R package. So feel free to assist here. This is an open source project. So what this allows us to do is, instead of just visualizing kind of one state at a time, I can kind of layer these things over top of each other and further customize how I want my visuals to look. So if you are a phylogenetics person or an infectious disease person and you want to use these visuals for a publication of your own, feel free to use StrainHub. And if you want really in-depth customizations, you can now use the StrainHub R package. So using the same information that I've been showing this entire time, now I can overlay host information on top of the location information, whereas I couldn't do that before. So what you see here on the screen is that you still see that same kind of network view where the central location here is kind of the hub, but you can see where the transmissions are, bat to bat, bat to bat, so on and so forth. But then you see bat to bat and then bat to pangolin. You see some sort of a pangolin transmission here, and then bat to human from Yunnan back to Hubei. And this transmission here is the bat to human. And this allows you to kind of see some information that isn't exactly clear from a complex phylogenetic tree. And I know that the tree that I'm using here is only 24 taxa, but as things start growing, then these networks really make things a lot easier to see. Okay. And with that, I will turn it over to questions.
Thank you so much. It is a shame that we don't have people applauding in the end. It was an awesome presentation. That's the only thing that I don't like about Zoom, that I'm not traveling to attend to this meeting. I will start with the first question that I saw. It's from Leandro Silvestre Gomes. Again, people, I'm very sorry if I'm butchering your names. He asks, what are the tree and geographic data supported by StrainHub? Nexus.tre, Excel, also from the same person. As planned for use in epidemiology in biogeographical sense, StrainHub prioritizes dispersion.
Yeah, so when you go to the site to begin with, you have a couple of different options here. The parsimony files can be PHYs, the Newick format files here. For beast phylogeography, you can also include, this is kind of more maximum likelihood based methods. So here you can use Nexus files. And then when you go through and select for the beast phylogeography, then it also allows you to change some thresholds here, so you can kind of trim the tree based off of particular probability or posterior probability thresholds. And then, so those are the input types there. The other is for neighbor joining. I mentioned this before, but the you can bring a sequence file. This can be an aligned FASTA file, and that multiple sequence alignment, we will create a neighbor joining tree for you, and then you can choose the bootstrap value. Just know that this is an open source project, so, and this is being hosted on a kind of a low powered, cheap server. So if you want to create your own trees using the software and you ramp this up to, like, let's say 100 or even farther, it may time out before it finishes, but you can run StrainHub locally from your own device, and you can, if you have a beefier device at home, you can kind of get away with more. As far as the metadata and geodata files, it's just CSV, and it's a pretty simple format. For metadata, the first column has to be accession, and it has to match the tip labels of the actual tree itself. But then the other columns can be whatever you want, whatever bit of metadata information. And then geodata just has to match one of the columns from metadata. So my geodata file here is at the state level, and then my metadata includes state. So really the geodata is matching the metadata, and the metadata is matching by accession number back to tree file. But it is quite flexible with what you want to show by. And the geodata file really is just the whatever column you want to use is the first column, which could be country or state or whatever, and then the latitude and longitude columns, and that's it. And in fact, I can show you that here. This is what that looks like.
Colby, if you were done answering the questions, thank you. I think you completed your answer. The next question is from one of our panelists, Dr. Anderson Brito. He asks, can network data be downloaded from StrainHub to serve as input to other software, like, for example, Cytoscape? Also, can we input trees in JSON format? JSON spells J-S-O-N. I'm not sure if that's how I should say it in English.
Yeah, absolutely. For JSON files, we do not have that ability to open JSON files today, but if that is a feature that people want added, feel free to make a request in the GitHub repository. But as far as downloading the files, you can download the networks themselves as an HTML and a PNG file. Very soon, I will also include the ability to download the file as an R dataset, so that when you're looking at the StrainHub R package, you can start from here, download the file, and then further customize if you so choose. That is not in the web app yet, but that will be there soon.
Okay, we have a few more questions. Next is from Lucilla Hondson. She asks, is there a limit to the file sizes that can be uploaded?
There is not a hard limit, so to speak, as far as the file size themselves. However, because we are hosting this on Shiny Apps, if for those of you that are familiar with this is a very low powered Amazon Web Service instance here. You can run StrainHub locally, and which relinquishes a lot of that those limitations here. So, for example, you can visit the GitHub repository and run the web application of StrainHub locally, and then it will use the resources of your machine. We have tried this with fairly large trees, and it doesn't have a problem, but we do know that it can take a while to do the processing. So just keep that in mind. If you use StrainHub.io and you're using a very large file and it doesn't complete, like the server times out before you get your result, then I would just suggest you run the kind of the home edition, so to speak. Or the other version is that you can use the R package version as well.
Thank you. The next question. Is there an option to use geographical coordinates, but not predefined areas or localities?
I'm not exactly sure what the question is asking here. For the geodata file, you do need to have the data, the first column of the data match a particular metadata attribute. So for example, this could be state or country or region or whatever. And then you will have the and long information there as well, but it does need to match something in the metadata. So it's not some sort of amorphous thing. And the way that this is generating the map today is that it's using an average lat long of the geodata. So it automatically kind of goes to a particular region on the map. Though with the R package version, you can further customize the leaflet-based map to your heart's content. You can use whatever you like and overlay other layers. So for example, if you wanted to overlay other regional information or something like that, you can do that using the R package version, just not the web app.
So I think I can provide some input so we can answer this question better. I'm guessing here, but I'm supposing that the person was asking whether or not we could give the StrainHub information like latitude and longitude in numerical values. And are you saying that we could potentially do that if that information matched the metadata? So for example, if we have a map and the map has information about latitude and longitude, then yes, you would be able to do that, give that information to StrainHub. Is that correct?
Correct. So for example, the format of the geodata has the column, the first column is actually very flexible, but whatever that first column is has to match the particular state that you're visualizing for the network. So if you take the tree and you summarize the tree down to the country level, then your geodata information that you upload should be at the country level with lat and long information. But if you summarize the tree and generate a network that's at the host level, for example, then you can't really generate a geodata file using the web app because it's kind of at a different level of summarization. But yeah, it is quite flexible. It just has to, whatever it is, whatever you input as the geodata just has to match the feature that you're summarizing or generating the network by, whether that's country or state or region or whatever.
So in the file you were showing us, in the column state, which is the first column, those names are arbitrary. You just chose them so that they would represent the latitude and longitude in columns 2 and 3, correct?
They are not arbitrary. They are actually belong to a feature in the metadata. So if I show you the metadata here, you can see I have accession numbers for the individual taxa that are on the tree. And then I also have, for each accession number, I have a state here. So Hubei, Xinjiang, for example. And then for each of these states, I have then matched that information in the geodata as well. So they're not necessarily arbitrary, but they match up with something that's in the metadata. So the metadata is really at the individual accession level, and then the network gets generated from that, whatever you select, and then the geodata then can take that metadata and then overlay it on a map based off of the lat long information that you include.
Thank you, Dr. Ford. I think we covered that. I have a final question before we move on to the next speaker.
Okay.
And you mentioned that StrainHub could be downloaded locally. And my question is, would it be something that is very hard, or would you say that a researcher that is not super familiarized with programming would still be able to install that and run in a local machine in his or her laboratory?
Yeah, if you use, if you're at least marginally familiar with R, you can download and get this to run locally. So if you clone the repository or download the entire repository itself, the app actually has an R Studio project file, so the StrainHub R project, that will literally allow you to just open the file in RStudio and click Run App, and then you'll see this exact same view on your local machine. It's just going to use your local machine's resources rather than the servers. So it's actually quite easy as long as you are at least somewhat familiar with opening up RStudio files.
Thank you. I think that this ends this session. Thank you very much, Colby, for coming here and being part of this.
Yeah,
it's
absolutely.
Awesome.
Thank you.
Susanna, your turn.
Okay, so for our next speaker, we are accepting questions in Spanish and English. And our next speaker is, just let me 1 minute, Daniela Mónaco from Emory University. And she is going to talk us about understanding SARS coronavirus 2 quasi-species via long-read next-generation sequencing. Thank you, Dr. Mónaco.
Thank you, Dr. Mónaco. I'm sorry to interrupt.
Yes.
It's 5:09 p.m. UTC -4 now. We will wait until 5:15 for you to start while we just check to see if the streaming is okay and if you are capable to share your video and your slides. So if you want to go ahead and try sharing your first slide with us, just so we can guarantee that everybody can see that, please.
Yes, no problem. Can you see that well?
It's fantastic.
Yeah.
Susanna,
Perfect.
everything okay in your end?
Yeah, it's perfect.
So I'm just going to ask for us to hold for 5 minutes. Meanwhile, if anybody has any technical issues or anything that they want to speak with me, please send me a message. I will mute myself, but I am online. And Dr. Mónaco, I will give you a 1-minute warning before 5:15, just so you don't lose track of time.
So you let me know when to start?
Precisely.
Okay. Thank you.
Okay, while we are waiting to start the next session, I have a few warnings, especially because sometimes we have people that are just joining in. If you are an attendee, we will receive your questions using Zoom's Q&A. So please submit your questions there. Susanna, can you say again which languages Dr. Mónaco will be receiving questions, please?
Yeah, Dr. Mónaco is receiving questions in Spanish and English. So please just send those questions to the Q&A chat.
Thank you. This is also one of the talks that's going to be streaming in our YouTube channel. Other panelists can also submit questions, but they cannot use the Q&A. So please, other panelists, if you have a question, just submit them using the chat. Dr. Mónaco, you will be ready to start in 1 minute.
Tengo 23 personas. Somos todos argentinos. ¿Qué dijiste para leer juntos?
It's 5:15. You may now start, Dr. Mónaco. Thank you very much.
Thank you, Denise. So thank you very much to the organizers for giving me this opportunity to share this very much ongoing work. And of course, to all of you on the other sides of the screens that are joining me right now. So today I'm going to be talking about the work that we are doing on trying to understand SARS-CoV-2 quasispecies using long-read next-generation sequencing. Sorry, I'm trying to change the slide there. So just to get started, I wanted to talk about a little bit about what we know so far about SARS-CoV-2 evolution. And we're very much aware of how this virus first emerged in Wuhan, China at the end of last year, and how since it has become a pandemic with that has caused over 16 million infections at 600,000 deaths worldwide. Since the beginning, a lot of effort has been put on sequencing the virus, and over 70,000 complete genomes have been collected that have allowed us to reconstruct the genomic epidemiology of the virus and have uncovered 5 different clades that are showed here in this phylogenetic tree using approximately 20,000 genomes from all over the world. And this work is from the NextClade website. Nevertheless, we see that the diversity of SARS-CoV-2 is very low compared with other RNA viruses, but rather similar to other coronaviruses. And this is most likely due to the proofreading capacity of the NSP14 protein. Rather striking evidence of the evolution of SARS-CoV-2 comes from a recent report that has shown the emergence of this D2G mutation in the spike protein that happened in February in Europe and was able to replace the wild-type variant coming out of China and become the dominant pandemic form. Though in this report, they associate this increase in the prevalence of this mutation with an increase in effectivity of variants harboring this mutation. Others have also proposed that this may be related also to a greater exportation of cases from Europe when we compare with exportation of cases from Asia. But overall, work has focused on identifying variants at the population level. And no study has really evaluated the diversity of the virus at the individual level. So in our study, we're trying to evaluate what's the intra-host viral diversity of SARS-CoV-2. And we think this is important because this might have implications for disease severity. So this is an schematic of the workflow that we did. So we started with 7 nasopharyngeal swabs that we obtained from Grady Memorial Hospital here in Atlanta, Georgia. This is one of the main hospitals in the city. And we also had a viral stock from an isolate from Washington here in the US also that has been used for in vitro studies, so has been used in several passages. And what we did was extract the RNA and do an RT and nested PCR of 3 overlapping segments that cover the almost complete genome of the virus. And we did this in 4 replicates in order to try to better capture all the minor variants that might be present in the samples. Then we did a library preparation, and in order to be able to identify the variants that belong to each of these amplicons, we barcoded these amplicons using barcoded adapters. So basically, each of the amplicons had a different barcode, and in that way, we will be able to identify which variant came from each amplicon, from each replicate. Then we proceed to sequence these using smart sequencing, using the SQL2, and we did the data analysis, basically mapping and haplotype calling in collaboration with Sequogenics, which is a company that's a spin-out of Emory that has developed algorithms that allowed for this mapping and haplotype calling in smart sequencing reads. So this is an schematic of the analysis of the smart sequencing data. So we start with raw uncorrected subreads. We started with 166 gigabytes of raw data, which corresponded to 6 million sequencing reads and 37 million subreads, with an average read length of 100 KB. And in this figure, you can see how these reads were distributed into the 3 amplicons that cover the complete genome of the virus. So using these subreads, we demultiplexed the data using this tool called Lima into the 87 datasets, which correspond to basically, and here I'm showing an example of, for example, patient 1 with segments 1, 2, and 3, and each of the segments with 4 barcodes. So for example, for patient 1, there's 12 datasets. And that happens for each patient and for the viral stock. And in this figure, I'm showing the coverage that we have for the genome. And here is segment 1, segment 2, and segment 3. And you can see the coverage is very homogeneous. And we have these spikes in coverage that corresponds to the regions where the sequences overlap. And then using these separated data sets, we perform the variant calling using Sequogenics tools that are available on the cloud. And then we analyze the sequences, performing phylogenetic analysis using Mr. Bayes, and then mutational analysis using the tools that are available online, thanks to NextStrain and Lanel. So starting with the phylogenetic analysis, we constructed a single consensus sequence per patient, and we did a Bayesian tree using sequences from Wuhan, China, and also from the main hotspots of the epidemic here in the US, which are California, New York, and Washington, as well as all the sequences that were available online for Georgia. And as you can see here, the sequences shown in red, most of them of our patients cluster with the available sequences from Georgia. And here I'm showing the mutation calling and clade assignment for these consensus sequences. You can see most of the sequences were of 19B clade. And interestingly, only 2 of the 7 patients have this D2G mutation that was previously reported as being the predominant form of the pandemic. And as expected, it has the accompanying mutations that usually are present when these mutations are present. So going into the identification of the variants, you can see here all the 3 segments and each of the patients for each of the segments. And you can see in segment 1, we were only able to identify 2 segments, 2 variants for this segment in 2 of the patients. But in the case of segments 2 and 3, we were able to identify more variants between 2 and 4 for each of the patients. And thanks to using coverage, we were able to calculate the variance frequency. So I'm here showing an example for patient 1. So we have, for example, fragment 1 and the 4 barcodes that correspond to the 4 amplicons. So we calculated a frequency based on the numbers of reads that constructed each of the variants, and then calculated a median frequency based on the 4 amplicons. And what I wanted to show here is that finding these variants was very much consistent, usually, between the 4 amplicons. As you can see here, each of the variants was found in each of the amplicons. But there were cases, for example, as you can see here, for variants that were very low prevalence, especially when prevalence was below 1%, that not all of the variants were showing up in all of the amplicons. And here I'm summarizing the frequencies for each of the variants that I was showing in the mutation plots. And in red are highlighted all the frequencies that are above 5%. So what we find is that there's 2 patients where we can identify variants that have more than 5% frequency. So there's patient 1 in fragment 1 that has two variants, one at 75% and one at 25%. And then there's patient 2 that has 2 variants at 50% each in fragment 3. Then all the remaining variants were of very low prevalence, many of them below 1%. And here I'm showing the mutations that define each of these minor variants. And what I'm showing here is that you can see we find both synonymous and non-synonymous mutations. And in some cases, the mutations were repeated between patients. But something that we were concerned with was with this low prevalence, if this could be related with sequencing errors. And we know that Paguayo errors are related with mutations being present in homopolymers. So as you can see in this last column in red, all of these mutations were not present in homopolymers. So we feel very confident that these mutations are real, and many of them are below this 1% frequency. And in this case, I'm showing here with the red arrow that these are the 2 mutations that were with higher frequencies, 25% and 50%. And interestingly, one of these mutations is non-synonymous. And then going into what we see when we look at the viral stock, you can clearly see that we see much more variation in the viral stock, especially in the segment 1. And when we compare frequencies calculated for the patient samples and the viral stock, you can see there's much more cases of variants with higher frequencies. So to conclude, we've shown that we found 2 of the 7 patients where we find additional variants that were present with relevant frequencies. And in the rest of the patients, we were able to find in each case additional variants, but in most of these cases, the frequencies were very low. In some cases, we think this may be related with sequencing errors, but in some cases, these mutations are very well supported by the analysis. In looking at the viral stock, we also see that diversity in this viral stock is much greater than what we see in vivo, especially in the segment 1, which contains ORF1A. And this demonstrates that the virus has the potential to diversify, but is clearly subjected to significant fitness constraints when it's transmitted in vivo. And for future studies, we are expanding this to a greater number of patients, and we are trying to evaluate if there's correlations between these different variants that we are observing and the severity of the disease in these individuals. So with this, I would like to acknowledge everybody that was involved in this study, people at Emory University, people that at Grady that supplied the samples, and also the viral stocks, and people from Sequogenics that help with the sequence analysis. And with that, I will be happy to take any questions.
Thank you, Dr. Mónaco. Well, I have some questions from my part. First is, have you find any association between those variants and the status health of the patients that you analyze in this study?
So we are trying to look at that. We haven't received yet the information about, for example, what's the story of, like, clinical story of these patients. We're trying to gather this data. But of course, in the following studies, we are trying to start with patients that we, have these data available so we can better understand this.
Okay, great. Thank you. And also, have you, do you know if there is an implication or probably effect of those mutations on replication activity or infectivity of the virus?
So we haven't really kind of done a very deep look at these mutations yet. This is very much a very recent analysis, but that's in the works. We're interested in this, and we've been looking at prevalence of these mutations in the population, thinking that that might be an indication of the fitness of these mutations. And for example, 2 of the mutations that were very low prevalence and that we think that might be related with sequencing errors were actually completely absent in the 70,000 genomes that are available. So we think that that's kind of a clear indication that might be, or a sequencing error or a mutation that's clearly not viable in vivo, right?
Great. We have one question from Dennis. He asks how important it is for us to understand the insertion in SARS-CoV-2 spike protein called polybasic cleavage site. Do you think that that could be a new insertion only in humans?
Which insertion? I couldn't understand, sorry.
Okay, I'm going to repeat the question. How important it is for us to understand the insertion in SARS-CoV-2 spike protein called polybasic cleavage site. Do you think that that could be a new insertion only in humans? Like the PBS site? Like.
Yes, I'm not really kind of familiar with that work. So I'll have to say I don't know.
Okay, perfect. And do you have that the diversity for this virus is overestimated, probably due to the sequencing errors?
Yeah, so that's a concern. We think that given the frequencies that we are reporting, we can't really say that we're reporting a great diversity, right? Because the frequencies are very low. So basically, we are just saying that 2 out of 7 patients we're seeing these variants, and it's just 2 variants that are present in each patient, right? So still the diversity is low, and that's what is expected for these variants, right?
Right, Dr. Mónaco, many thanks for this interesting talk. And thank you for your time. Now we are going to be prepared for a break of 15 minutes. So we are going back at 16:50. And then we will start the second part of this session with the Dennis Jacob Machado talk. So stay tuned for the second part and see you soon.